In this video, we're going to talk about the electron transport chain, and in some ways we need to answer the questions of, sort of what to do with NADH and the FADH2. Right, so we've made these molecules, we've made them by oxidation reduction, and what we've said is that if we take something like NAD plus and we convert it to NADH, that we've transferred energy over here, and so this molecule is high potential energy. Okay, so the question becomes, how do we get the energy moved then from NADH into ATP? All right, and we're going to do that by a combination of electron transport and chemiosmosis. Okay, so we're going to do electron transport, and then we're going to do something called chemiosmosis. All right, and so we'll sort of address these issues. All right, so let's look at the electron transport chain. And so we have electrons, which are our energy source, and so they're going to come from NADH, let's just say for now. All right, if they came from FADH, okay, so if they came from FADH2, perhaps they enter maybe down here. All right, it's just a way to think about it. So there's less energy associated with the electrons in the FADH2 than there is in the NADH. So what we're going to do then is a series of redox reactions where the redox, so we're going to oxidize our NADH, and the electrons are going to move from one location to another, and in doing so they will lose energy. All right, and so they're going to work their way down, and every time we see the electrons move, they go to a new electron carrier in the electron transport chain, finally ending up on oxygen to make water. All right, and so what we're going to do then is that as we release the energy from these electrons, all right, we're going to establish a proton gradient. All right, so it says proton motive force here, but you should be thinking of we're actually moving protons to one side of the membrane so that we can have a high concentration of protons. All right, so let's look at the electron transport chain. So here's NADH going to NAD plus. All right, so we're oxidizing the NADH as it becomes NAD plus. So we're removing the electrons, and they go to our first electron carrier. All right, we then will transfer those electrons to ubiquinone, and as we do that, we will move protons from inside the cytoplasm to outside of our cell. All right, so we're just going to move them across. Ubiquinone will take them to a second carrier, all right, which will then transfer those electrons to oxygen to make water, and we will pump protons a second time. All right, so this electron transport chain is shorter than you've probably learned previously, and it's just sort of a simplified version of what we would find in E. coli. Now, if we had FADH2, so if we had FADH2, its electrons would come in here at succinate dehydrogenase, all right, and so it would become FAD, and our two electrons would move on through. All right, so each time we transfer the electrons, right, so from here to here, that's a redox reaction. This became reduced, this became oxidized. Then we move those electrons to ubiquinone, so now this is oxidized and can receive electrons from the next NADH. Ubiquinone moves through the lipid layer, all right, and brings them here, and we then transfer the electrons to oxygen, and we pump protons a second time. Now, if we've established this proton gradient, all right, the question is what to do with it. And there's three common things. So one would be active transport, right? We've talked about this mechanism of transport where we use the proton gradient. The other is to cause flagella to rotate. So as the protons are allowed back in through through the flagella mechanism, causes it to spin. And then the third is the actual taking of H plus through ATP synthase, and this is the chemiosmosis. All right, so we take a high concentration of protons here, allow them to move to the inside. This causes this enzyme to spin, and it will combine, all right, and we're doing this for 3 ADP and to make 3 ATP, and we're using 10 protons. All right, so what we're going to do is we're going to take inorganic phosphate and we're going to combine it with the ADP to make ATP. All right, so let's diagram this really quick just so that we have a better feel for it. So if this is my membrane, all right, so what I'm going to do is I'm going to put my electron transport chain here, and I'm going to put my ATP synthase here. Okay. Now, as the electrons come from NADH, all right, we are going to then move our protons across, right? So we're going to establish a very high concentration of H plus on this side, okay? We will also reduce the amount of H plus on the cytoplasmic side, right? So if I write, so this is my cytoplasm here. When we take the 1/2 O2 plus 2H plus, right, and my electrons, okay, that will consume some H plus on this side. So we're going to have low H plus, high H plus. So now this is going to work just like osmosis. But we're going to call it chemiosmosis because it's a chemical. All right. And what we're going to do is we're going to move these H pluses through the ATP synthase enzyme, and it will combine ADP plus inorganic phosphate to make ATP. All right, and we call this overall process oxidative phosphorylation. That's the entire process is called oxidative phosphorylation. Okay? Now, we won't always have oxygen present, all right? And so in that previous example, we used oxygen, we used the electron transport chain. So we've got some other ways to drive anaerobic growth, so to provide ATP for cells in the absence of oxygen. The first being fermentation, all right? And what we do here is we regenerate NAD plus so that glycolysis can continue, all right? We refer to it as internally balanced with acceptor and donator of electrons, meaning that the fermentation process itself generates the electron acceptor. So you don't have to have an external electron acceptor. And it's going to yield very little ATP, right? So very little energy is going to be, and so we're going to get 2 net. Okay, so that's net. Now, anaerobic respiration, all right, is just like the entire process that we just went over for cellular respiration with one change. All right, so we do glycolysis, transition step, citric acid cycle, electron transport. The only thing that changes is that rather than using oxygen at the end of the electron transport chain, we use something different like nitrate or sulfate. All right, so it'll yield less total energy than oxygen, but more than fermentation. How does the anaerobic respiration work? So if we think of that initial stair step, right, and down here at the bottom is where we have O2, right? So our electrons, right, our electrons are going to bounce down, and we will put them onto oxygen and we will generate water, right, as we go like this. Okay, now what we're talking about is the potential energy, right? So, and it decreases, and this is of the electrons, right? So the energy, and so we're going high to low. So let's put nitrate and sulfate on here, and so maybe the nitrate is here and the sulfate is here. And so what happens is the electrons don't get to fall as far, so they don't lose as much when they get added to the sulfate to make H2S, for example. All right, and they don't fall as far as oxygen when they get converted to N2 gas in terms of nitrate. All right, so because of the less energy is released from those electrons before they're done being transported, what that means is you generate less of a proton gradient, right? You generate less proton motive force. All right. Fermentation, all right, occurs after some manipulations of the carbon. And so in this case, we're going to look at two examples that occur after glycolysis. All right, so if we look, we start with pyruvate in both of our fermentations. So lactic acid fermentation, this is what occurs in your muscles if they go anaerobic. So here's pyruvate, it's 3 carbons, all right? And if you remember, in glycolysis, we generated an NADH, all right? So what we have to imagine is that if all of the NAD plus, okay, so if all of the NAD plus has been converted to NADH, you can't do glycolysis anymore, right? The oxidation reduction step cannot occur if all of it is in this form. So what has to happen is we need to regenerate the NAD plus so that we can allow for glycolysis to continue, and we continue to generate a small amount of ATP. Okay, so that's the fermentation. So pyruvate, if pyruvate accepts the electrons from NADH, notice NADH goes to NAD plus, so then the pyruvate will become lactate or lactic acid. All right. So that is an oxidation of NADH back to NAD, a reduction of pyruvate to lactate. The NAD plus now can go to glycolysis and get converted back into NADH. Now, another form, all right, that generates ethanol, the pyruvate is decarboxylated, right? So we remove a CO2 from it. That gives us acetaldehyde. Acetaldehyde is then reduced by NADH to NAD+. So NADH goes to NAD+, the acetaldehyde accepts those electrons and becomes ethanol, which of course can be consumed. All right, so that's fermentation, and what we have to think of is that on a per glucose, all right, per glucose, when you're fermenting, you get 2 net ATP. All right, for the anaerobic respiration where we use nitrate, sulfate, maybe you get 20, 28, something like that. So you get a lot more when you do anaerobic respiration. All right, so that should wrap it up.